One of the interesting things about our species is that we live socially. We live socially in a very specific way. We're not just in these amorphous groups, like a herd of cattle or a bunch of crocodiles. We live actually in social networks. Each of us has specific friends, and those people in turn have their own specific friends and other social contacts. And we assemble ourselves into these elaborate social networks, which is rare in the animal kingdom. We do it, chimpanzees and certain other primates do it, elephants make networks, actually orcas and dolphins will make social networks, we think sperm whales as well. It seems like multiple times over the last 90 million years, in very diverse taxa, we evolved this capacity to assemble networks. Independently, these animals evolved this capacity. And so this is clearly a behavior, a phenotype that is shaped by natural selection. And so the reason is why. It must be the case that the benefits of a connected life outweigh the costs. Because if every time I came near you, you lied to me, you gave me false information, or you stole from me, or you injured me, or you killed me, I would be better off living atomistically. We'd all be better off living apart from each other. What we really want to understand is the extent to which olfaction plays a role in human social relationships, non-reproductive relationships. So to what extent do the odors that we naturally emit send information about us, our trustworthiness, our friendliness, our cooperativeness, to other members of our species? And how do those people detect those signals and choose whether to befriend us or not? So we really want to understand the degree to which olfaction plays a fundamental role in human social relationships.
We know that social networks are a complex concept and a complex reality to understand. It is not enough to have the angle of a single discipline, such as psychology or sociology or biology or genomics, right? The beautiful thing of what we are doing here right now is that we are combining not only disciplines, but really big corpus of knowledge, hundreds of years of questions that we have had, but that we now have the tools to investigate a little better. So, for example, how does smell impact the ways in which we befriend a person? That's something that we're trying to look at right now. We have a team based in Copan that we have been working on for the past 8 years. And we have our team here in New Haven at Yale.
The Honduras team is one of the most hardworking, committed, and disciplined group of people that I've ever had the pleasure of working with. Being in the field and working with them and walking and doing these trajectories and going to houses and being there until 6 p.m. collecting data, seeing that level of passion and dedication and want to be involved in our projects and really want our projects to be successful is just it really shows their commitment to us and their commitment to the communities.
Bueno, los datos nosotros los recopilamos por medio de encuestas y hacemos el levantamiento de información con cada una de las personas que está participando en el estudio. Hemos tenido la oportunidad de hacer el levantamiento de esta información por medio de una entrevista o bien por medio de datos, ¿verdad? Que ellos pues nos brindan. Tuvimos la oportunidad, por ejemplo, de recopilar información cuando hicimos el proyecto de microbioma, ¿verdad? Que aparte de ellos brindarnos información, nos brindaron muestras, ¿verdad? De saliva, muestras de heces y algunos datos de salud. Entonces, esa es la manera de cómo nosotros interactuamos con las personas. Aquí estamos afuera de la casa donde usted está. ¿Quieren entrar a observarnos?
So we began the exploration of this idea about 15 years ago with work on the evolutionary biology and genetics of human social interactions. One of our first studies was a simple twin study, and we looked at same-sex dizygotic twins and monozygotic identical twins and looked at the networks that they form, and we found that the social networks formed by monozygotic twins were more similar to each other than the social networks formed by same-sex dizygotic twins, which suggests that there's something heritable about our ability to make friends and to create the networks and live in the networks around us. And we followed that up by looking at the genetic similarity of friends. And we found that actually, when people are free to pick anyone as their friends, among unrelated individuals, they pick as their friends individuals who resemble them genetically at the level of third or fourth cousins. In other words, these people are not your cousins, but the people you pick as your friends are like your kin. They have some kind of genetic resemblance to you. And in parallel with that work, we also began to look at the physiology of friendship. So for example, everyone is familiar with the idea that when you're with your friends, you just feel better. The kind of sense, that feeling that you feel better, your heart rate goes down, your sort of emotional state changes. And it turns out biomarkers, certain circulating proteins in your body can also change. And we found, for example, that this biomarker of stress called fibrinogen is more common in the periphery of social networks. That is to say, socially isolated people had higher levels of fibrinogen than more socially central people. Our bodies have been shaped, our minds have been shaped by natural selection to have this capacity to very quickly choose people who might be suitable friends for us, people we trust, people we like, people whose company we enjoy, people who we would like to be friendly with. So there must be some underlying biology to this. There must be some evolutionary shaping, some physiology, some means by which we can actualize this sensibility of like at first sight, or click friends, making friends just immediately. So I think, either consciously or subliminally, we can smell people who would be suitable friends to us. And that this capacity, which has been shaped over hundreds of thousands of years as our species evolved, actually lies behind our propensity to make social networks in the first place.
The social networks that we build are not separated from our particular biology. We have research in our lab that has shown that our microbiome affects the way in which we befriend people and the ways in which we relate to others.
So our research is trying to understand how friendship can be seen as a vehicle for transmitting, for example, specific bacteria. In our case, we are not trying to focus just on specific bacteria that are pathogenic, but also to commensal, so bacteria that are commonly found in the human gut microbiome.
We did a study in Honduras of 18 villages and nearly 2,000 people. We carted liquid nitrogen into the field, in the jungle, which was not easy. We persuaded these 2,000 people to poop in little cups, which was not easy. And then we also photographed these whole villages and mapped their social networks, which also was not easy. And then we sent all that data back to the United States and processed it, and were able to study the extent to which people who were friends with each other came to share similar microbial strains of the bacteria in our guts. In other words, we were better able to predict who your friends were based on poop samples than based on your similarity on axes such as religion or education or age and so on. And in fact, as part of that study, we mapped the networks and we found that there were sort of clouds of bacteria, human beings, float in the microbes of their friends. That there were clouds of microbes in clusters of friends within social networks, so that groups of interconnected people in one region of the network came to share microbes different from the microbes that were shared by another group in a different region of the social network. And all of that work on the genetics of friendship, the physiology of friendship, the microbiome of friendship, set the stage for our new work in volatile organic compounds and chemosignaling that are part of friendship.
Specifically, we are investigating the chemical profiles of human skin secretions and their potential to signal cooperative tendencies. This involves sampling semi-volatile organic compounds and using technologies like gas chromatography, mass spectrometry to analyze them. And additionally, we are examining how olfactory cues such as scent similarity among friends may influence social interactions.
People emit volatile organic compounds from their bodies. These compounds relate to the diet of the human beings, the genetics of the human beings, the environment of the human beings, the microbiome of the human beings. Many factors go into creating our distinctive odor.
If our findings confirm that chemosignaling play a role in social bonding, it could fundamentally change how we understand relationships. It would highlight that friendships are not only influenced by shared experiences and choices, but also underlying biological mechanisms, such as our ability to detect subtle chemical cues.
Human social networks are this extraordinary thing. They're these baroque, ornate structures that we naturally make. They exist in the world. They're deeply interesting objects. They're phenomena that we might want to very deeply understand. To the extent that we can understand why human groups work well or poorly, and to the extent that this might have something to do with the kind of chemosignals that we send to each other, this might even provide a kind of set of diagnostic or therapeutic opportunities for us. We might be able to literally understand why some groups of people don't work well together, or why some groups of people are all depressed, for example. Maybe it has to do with the chemosignals they're sending each other. And understanding that, understanding the role of olfaction in human beings and in their social behavior is absolutely a scientific frontier right now. So, our gaze is firmly fixed on that topic. We, of course, continue to be interested in many other aspects of human social interaction, but let me just say, some part of our gaze is definitely fixed on that topic right now.